Hi, in this chapter we are going to see about the muscular tissue. So previously we were talking about the epithelial tissues and connective tissues, and I hope you get a very clear picture of those tissues. So in this session we are going to talk about the structure of the muscle tissues, about the muscle fiber, about different types of muscles, and then I'm going to talk about some physiology sessions here, because definitely we want to talk about how the muscle contracts. Am I right? So let's dive into the session. So here is the cross section of a muscle. For example, say a biceps. So here you have the muscle fiber, or you say the muscle cells. The reason why we call the muscle cell as a muscle fiber, because the muscle fiber is so elongated and it is very different than a typical cell. That's why we call it as a muscle fiber. So the muscle fiber is surrounded by connective tissues, because if one part of the muscle group is contracting, the force generated can be transferred to the other parts of a muscle. That's why connective tissues are so important in the muscles. So the entire muscle is covered up by the connective tissues, which is called the epimysium. And the group of muscle fibers here forms the fasciculars, or you say muscle bundles. And the connective tissue surrounding the bundles is called the perimysium. And in between the muscle fiber you have some connective tissues which is called the endomysium. Now what I'm going to do is take a muscle fiber, I'm going to compare with a typical cell that you learned so far. Let's see how different a muscle fiber is. On this side I have taken a typical cell, and this side I have taken a muscle fiber. There are at least 5 important points you should know. The plasma membrane or the cell membrane in a typical cell, here it is called the sarcolemma. The sarcolemma has some invaginations which we call it as T-tubules. We'll discuss this in a while. And the second point is a smooth endoplasmic reticulum. And recall the important function of ACR we discussed in the previous chapter. It is lipid and steroid synthesis and detoxification of drugs. Here the ACR is modified to form the sarcoplasmic reticulum, and the important function of the sarcoplasmic reticulum is to store calcium inside it. And this calcium is important for muscle contraction. We'll discuss this in a while. Number 3 is a cytoskeleton. We already discussed about this in the previous chapters, that the microfilament is going to provide the structural support. Here the microfilament, that is the actin, is abundant here, and it interacts with myosin and produces the muscle contraction. And the last 2 points is about the nucleus and the mitochondria. Here there is a single nucleus which lies in the center of a cell, and the muscle fiber, the nucleus, are multiple and lies in the periphery. There is a small difference between the skeletal muscles or cardiac muscles or the smooth muscles in this point. We'll see this in the end of this session. What about the mitochondria? It is few here when compared to a muscle fiber. And here the mitochondria are abundant because the muscle fiber needs energy throughout the day. Okay, here is a picture that shows up the sarcolemma here, and there are some indentations that leads to a tube-like structure which is called the T-tubule. And this T-tubule extends deep inside the cell, and when the action potential traveling down the membrane, this T-tubule helps to take the action potential deep inside the muscles quickly. On both the sides of the T-tubule or the sarcoplasmic reticulum which stores the calcium. So when the action potential reaches the T-tubule, this is going to open up some channels in the sarcoplasmic reticulum, and the calcium pours into the cytoplasm and the muscle contraction begins. We'll see about the muscle contraction in a while. So one more point. Triad means in the center you have a T-tubule, on both the sides you have sarcoplasmic reticulum. If it is a dyad, in the center there will be a T-tubule, On one side there will be sarcoplasmic reticulum, because there is a small difference between skeletal muscles and cardiac muscles in this point. I'll tell you later. Let's move on to see an important histology image of the muscle fiber. So as I said, the muscle fiber is very long when compared to a typical cell. It is elongated. So each and every muscle fiber is long and cylindrical. You can see a lot of lines like this throughout the muscle fiber, like a nodes in a sugar cane. And this is the Z line, and in between the Z line the structure is sarcomere. So we can say like you have many sarcomeres arranged in series. Alright, so next image I'm going to take the EM of the sarcomere because that is a very important image. Okay, so this is one Z line, this is another Z line. In between the Z line you call it as sarcomere. If you analyze the sarcomere, you can see alternate bands of light zone and dark zone, light zone and dark zone, and so on. So alternating bands of light and dark bands. And this band is called I band, light band, and this is called dark band, A band. The I band contains actin and A band contains myosin. Now you can understand why this is light and why this is dark. Because comparatively, myosin is way thicker than the actin. How to remember this? Let me give you some tips. Light, I band, dark, A band. Note those second letters. And in the light band you can see actin. Actin. Actin is very thin. Actin. I hope this helps you. What is this line? Z line. And this is the sarcomere. In the middle of a sarcomere, you have the M line. M for middle. And this M line is present in a zone. You can see some light areas on both the sides of M line. And this zone is called the H zone. And this completes the EM of the muscle fiber. Now I'm going to take the thin filaments, that is the actin, and then the myosin, and explain it to you. And then we are going to see how the muscle contracts. Okay, so here is the actin filament. This consists of two subfilaments that are twisted against each other. And this actin filament contains actin globules. And in the actin globules, you have an area which is called the myosin binding sites. Here is where the myosin head binds onto. Let's see the text. There are two chains containing actin globules, and it contains myosin binding sites that interacts with myosin. There are two other proteins associated with this actin. Number one is a tropomyosin, the other is a troponin. We'll discuss about the tropomyosin and troponin. So here is a tropomyosin, a belt-like protein that is associated with the actin. This is going to cover up the myosin binding sites when the muscle is in resting state. And this troponin consists of three components. This is going to stabilize the structure. One component binds with the tropomyosin. The other component is going to bind with the actin, and the other component is going to bind with the calcium. Let's see the text. So during resting state, tropomyosin covers the myosin binding sites in actin. And number two, the troponin has three components, T, I, and C. This T is going to bind with the tropomyosin, and the I is going to bind with actin, and C is going to bind with calcium. And this component is very important. Now we'll see about the myosin. So here is a myosin molecule. You have two components, tail and the head. So myosin has two components, the tail and the head. And the head of a myosin has an important enzyme which is called ATPase. And this ATPase is going to cleave the ATP molecules and gets its energy. And this myosin is going to bind with the actin when tropomyosin uncovers. This point you will clearly understand in the next slide. Now we'll discuss how the system really works. This is going to be very interesting. Okay, so this is the sarcolemma and the T tubules here, which are nothing but the indentations in the sarcolemma deep into the cells. And here is a sarcoplasmic reticulum. This is going to store calcium inside. Under resting state, this channel is closed. And here is a Z line to where is attached the actin filament. This is thin filaments, and the thick filaments where myosin is present. Under resting state, the tropomyosin covers up the myosin binding site and the myosin head cannot attach to the actin. Okay, now the action potential is traveling down the sarcolemma of the muscle. And the T tubules is going to take the action potential deep inside. And this action potential is going to open up the channels so that the calcium is poured into the cytoplasm. And this calcium is going to bind with the troponin C. When the troponin C binds with the calcium, the tropomyosin is going to uncover the myosin binding sites. Now the myosin head goes to bind with the actin. Note that the myosin head has ATPase enzyme that is going to cleave the ATP and gets its energy and flex the neck. So this actin overlap against each other in this direction. And the Z line are pulled close together like this. And the muscle contracts. So this is how the muscle contraction happens. Now you have to know about some important difference between the three types of muscles. So there are three types of muscles. One is skeletal muscles, smooth muscles, and cardiac muscles. First, see these pictures. So you can see some striations here. You can also see some striations here. And these striated muscles are, this is skeletal muscles and this is cardiac muscles. And the non-striated muscles are smooth muscles. So if the striations are present, it can be skeletal muscle or cardiac muscles. If there are no striations, it is smooth muscles. Fine. How can you differentiate between the skeletal muscle and the cardiac muscle? 2 points. Look at the nucleus. The nucleus lies at the periphery. The nucleus will be centered here. And also, look at the branching pattern of a cardiac muscles. It's very specific to cardiac muscles. The smooth muscles, you can see the spindle-shaped cells here. And the nucleus will be in the center of the muscles. Look at this image. The skeletal muscles, you can see the nucleus will be in the periphery. And the smooth muscles, you can easily identify the spindle cells here. And the cardiac muscles, you can easily identify the branching pattern of the muscles. And the nucleus will be in the center. Let's summarize using a table. I'm going to read out this table. This table is going to be very easy because we have discussed almost all the points here. So in skeletal muscles, it is long, cylindrical, unbranched. Cardiac muscles, short, narrow, branched. And the smooth muscles are spindle shaped. The skeletal muscles, it's voluntary, it's under our will. And the cardiac muscles and smooth muscles are involuntary, it's under autonomic nervous system. And skeletal muscles you can see, for example, in skeletal muscles, esophagus, diaphragm. cardiac muscles in the heart, smooth muscles in the blood vessels and organs. What about the striations? Skeletal muscles, striations are present. Very poor striations in cardiac muscles. And striations are absent in smooth muscles. Remember the pictures. And the nucleus here are multinucleated and peripheral. Nucleus is single and center here in the cardiac muscles and smooth muscles. What about the sarcoplasmic reticulum is in triad? Here it is dyad, and here there is poor development of sarcoplasmic reticulum. About the intercalated disc is very specific for cardiac muscles. If you wonder what is this intercalated disc, nothing but a transverse line between each cell of a cardiac muscle. And one important point about this disc is where the gap junctions are present. We already discussed a lot about gap junctions when we did the chapter 3 cell junction chapter. What about the regeneration? When talking about the regeneration, the smooth muscles regeneration is seen. For example, say a wound healing, you can see the smooth muscles regenerating, but absolutely not in cardiac muscles. Regeneration is not seen in cardiac muscles. Once a muscle is damaged, it is damaged. The skeletal muscles, regeneration is seen, but it is limited. I hope this session is very interesting and useful for you. So now you get a very clear visualization of the muscle. Just imagine yourself how the muscle contracts. Next chapter we are going to see about the nervous system. I hope your imagination about the human body is slowly improving. When you finish this course, you will be absolutely ready. See you next chapter. Thank you.